two quick comments. This, I think, is a really appropriate approach to thinking about immigration, and happy St. Patrick's Day, about these other illegal immigrants who happen to be here. I also was looking at my bag that I bring all of your exams over here with, and noticed it's actually from a meeting that I went to in Dublin in the last century, which is kind of frightening. So again, today is all review. You can hide your clickers. I didn't even install my piece here. I will be uploading all of the scores this afternoon. I'll be trying to do that, just your total scores, and probably wipe out all the other ones. And that's the only one which really makes a difference. Yes?
One question, when I was looking for grade changes, there were a couple that were listed slash out of 100.
Yeah, so some of them were listed slash out of 100. You mean we didn't have 100 clicker questions that day? No, that's a D2L talking to iClicker thing. You can completely ignore that. Yeah.
When do we know what the scores normalized to?
For the grade, I will do that today as well. I'll post that when I put them up. Okay. Other, again, exam Wednesday, normal time here. More questions that come up that we don't talk about today. Please send me emails, and I'll be posting answers to all of those on D2L in the discussion section. And I will also be posting the recording of this, assuming that I remember to save it after class. So let's get started. Again, feels like forever ago that we actually started doing this. Really started talking mostly about proteins. At the very beginning, how proteins get put together. Usually, most proteins don't have the right specific fold or three-dimensional structure, whereas it comes off the ribosome. They need help, and the main thing they need help with is not interacting with the wrong other proteins or the wrong parts of their own protein. So the two different ways that this happens, two major ways that this happens, is either through the HSP60 or GRO-EL, GRO-ES system. GRO-EL, GRO-ES is bacteria. HSP60 is general for all of these, but is mostly used in eukaryotes. The incorrectly folded protein gets put into this box. It gets shaken around with ATP, and eventually you have the right one that comes back out. This happens after the ribosome, while you're getting translation of your polypeptide. You have the other set of machinery, the HSP70 machinery, and 70 and 60 is just the relative size on those STS page gels that we looked at just a couple of lectures ago. So that's where all of those numbers come from. Here is basically everything else that we talked about in this section of the course. Right now, again, this is the folding process after you've made your protein getting put together. Of course, if it doesn't get made properly, it gets degraded through the proteasome process. But then, again, the rest of what we've talked about, with the exception of the methods right at the end, is really all about this transcription process going from DNA to RNA. Starts out, of course, as a pre-messenger RNA with all the introns left in, no cap, no tail, etc. That processing, which we also talked about mostly last time. Then we get that sent out into the cytoplasm. It gets translated, etc. All of these steps are regulated. The vast majority of regulation has to do with this initiation of transcription here, which is, again, not surprising because it's the very beginning of the process. However, it takes a while, particularly if you're thinking about a eukaryotic system, because you need to do capping, elongation, splicing, etc., until we finally get to our protein at the end here. This is a classic example of an image that I think I did a bad job of explaining the first time around. So this has to do with actually what we talked about on Wednesday. Very high throughput sequencing, being able to get very large numbers of sequences in very short periods of time. This particular case, almost everyone is using the Illumina, those different colored dots that we talked about right at the very end of lecture. And basically what you do here is you take a cell or a tissue or something like that, get all of the RNA out of it, and sequence all of that RNA, and get many, many copies of all of the RNAs that happen to be in that particular sample at that particular time. These are really short. And again, we talked about this when we talked about the Illumina. All of these things are relatively short sequences. Then, in your computer, it just lines all these sequences up. Every little dot here, starting from the bottom, moving up to the top, is one sequence. And the computer can line all of these sequences up with each other and says here, and this will literally be thousands of sequences. The y-axis here is thousands of sequences that are found in an embryonic stem cell that match to parts of the beta-actin gene. So this is what this is supposed to represent. Each of these, basically, these are histograms of looking at all of the sequence data, which are here. So that, again, I think I did a crummy job of explaining that. But in terms of people looking at what genes are being expressed in any given cell type, any given tissue, any given time, this is really the technique that most everyone uses these days, called RNA-seq or RNA sequencing. So short sequences, line them up with a genome, see what you see. Two-dimensional gel electrophoresis, which we talked about the methods on Monday, separating based on relative molecular mass. Weight here shouldn't be, this is relative molecular mass, with STS-Page. So this is literally STS-Page, which is done here. But each of those STS-Page, what would be a horizontal band, has now been separated based on its isoelectric point using isoelectric focusing. So each of these dots here represents a protein with whatever modifications it happens to have. And we talked about this particular example over here. This is all one protein that's got multiple different isoelectric points because it's got multiple different numbers of phosphates that are attached to it, which is going to give that a different isoelectric point. So that's, again, the main thing that I wanted to talk about here, just to go over those again. This is basically exactly the same as, 3 slides ago, I guess, where we had this vertically. Now we're just looking at it horizontally. Regulation of gene expression goes all the way from transcriptional control to the activity of your protein. Transcriptional control is how you turn a gene on, you turn a gene off. This is the really fundamental process, but it takes a while before that transcriptional initiation eventually gives you an active protein. If you want to turn things on and off really quickly, you want to have it at this protein activity control level. Yeah, sure.
Will a protein remain inactive but not degrade in the cytoplasm?
Okay, so the question here basically, will a protein end up being active or inactive, just going to hang out as an inactive form in the cell? And of course, when we say a protein, it could be any protein. But it's certainly true for many proteins, and this particular example is one that's getting phosphorylated. So in the unphosphorylated state, it could be very stable, and then it can become phosphorylated really quickly. There are other examples that we talked about when we were talking about the cyclin-dependent kinases, when we talked about replication initiation. Those are very unstable. So it varies. But if you're thinking about having something that you can turn on or off really quickly, you're going to want to have that off form be pretty stable and hanging out inside the cell. And that's true a lot of cell biology, signaling, so on and so forth. So for the most part, yes, they are pretty stable, even when they're non-functional. Okay, again, another example of how I messed up, and everyone believed me when I messed up, and a few people called me on it later. So this has to do with DNA binding proteins, and particularly DNA binding proteins that you find in bacteria. They've got a classic helix-turn-helix DNA binding motif. So blue helix, little gray turn to another helix. This red helix is the recognition helix, has specific base pair interactions in the major groove of DNA. And since DNA, B-form DNA, does one turn every 10 base pairs, if you've got a recognition helix with a sequence here, 10 bases later, it's going to be here, which is 3.4 nanometers, not 34 nanometers. So that was the mistake I made in the clicker question, and also in the lecture. So it's 34 angstroms, which is 10 to the minus 10 meters, but 3.4 nanometers between major grooves. And it's just 10 base pairs. And what that means is these proteins can sit down on one side of the DNA. And it's a lot easier if you have these dimeric proteins, very common in eukaryotic systems as well as in bacterial systems, that can then sit down, again, one full turn of your B-form DNA separated. Example of that is this example for lambda control of repressor and other proteins. It's one of those transcriptional activators. It's important for positive feedback loops in lambda. It binds to this particular sequence, again in the major groove of DNA, 5' AACAC. That's one part of your dimeric protein. The other part binds to exactly the same sequence, 5' AACAC, in the opposite orientation. So this would be an inverted repeat sequence. And then you have a dimerization domain here, separate from the DNA. This is unfortunately, it's not a three-dimensional model, but these pieces are really quite separate relative to anything else. And so that's actually shown here. Here we've got our two recognition helices in lambda Crow. The dimerization is between these two beta strands here, well away from the DNA. So these half sites, individual monomers, binding together as a dimer. Dimerization motifs are pretty well known in eukaryotic systems. There are just a few of them. One of the really classic ones is the so-called leucine zipper. Again, this is a alpha helix, every 7 amino acids, a large hydrophobic amino acid side chain, very often a leucine, not always a leucine. But what that means is that these side chains can fit into each other, rather like a zipper process. When a leucine zipper is right next to a basic helix, often these will be DNA binding proteins. So they'll have two motifs, this dimerization motif, which is a leucine zipper, and then a basic helix that binds to the DNA. This is a little different than those 3.4 nanometer apart binding in two separate grooves. This is really one major groove which is right next to each other. So those half sites are going to be right next to each other instead of separated like we had in the previous example. Another dimerization motif is the helix loop helix. Unfortunately, not to be confused with helix turn helix. Turns are usually about 3 or 4 amino acids. These loops can be 10, 20, even sometimes more than that in terms of the numbers of amino acids here. But again, this is a dimerization motif. It's not a DNA binding motif. Why do we care so much about dimerization? Well, A, you can have these half sites where binding of one monomer to 4 or 5 nucleotides is not going to give you high enough specificity to bind to one or very few places in the genome. As soon as you make it twice as big, it gives you much more specificity. But also, if you have cooperative binding, which says once you have binding of one of the monomers, it also gets you binding of the second monomer, then you can have much more of a on-off kind of switch relative to protein concentration. And this is much more useful for regulating genes to have them turned on or turned off, as opposed to a slow progression, which is what you get if your dimers are always together, which actually basically behaving like a monomer. So dimers help in terms of getting you specificity, and dimers help in terms of getting you cooperativity of binding, which is this binary process right here. I wanted to bring up the EMSA because lots of people ask me questions about it. but it's also a nice example of looking at a chromatographic, separation. And so here, the green line represents your overall protein concentration coming off of your favorite column. In this case, it was a ion exchange column, and increasing salt concentrations as you move across it here. And each of the fractions that was taken off of the bottom of the column was taken, and a gel mobility shift assay was done with that fraction. And you can see in each of these fractions, at different salt concentrations, you have different DNA binding proteins that are interacting with your favorite piece of DNA right here. So this I wanted to use more as an example of a chromatography kind of experiment. We talked again way back at that point, we talked about DNA binding, about affinity chromatography. DNA affinity chromatography is a wonderful way to purify what kind of proteins? DNA binding proteins. And what kind of DNA binding proteins we talked about? Transcriptional regulators for the most part. So the way to do that is we've got two different columns here with two different matrices. What are matrices doing? They're slowing down what you want to hang on to on the column here. And so here, we're looking for these little red dots representing those specific, presumably enhancer binding proteins. Although in this case, with a palindrome, it's probably actually a endonuclease. So binding to the column here is going to be any kind of DNA binding protein. And so it's a whole mixture of different sequences here. Often people actually just have polyionic columns here. Heparin is a classic example of that. So generally negatively charged, so it kind of looks like DNA, something that binds to negatively charged polyions will bind to this column. So no DNA binding proteins, they come straight through. Your DNA binding proteins, which are a subset of the ones that you started with, presumably this is a cell extract that you got out of some kind of centrifugation. Then you take this mixture and put it over your specific DNA column. Again, the stuff that doesn't bind to it, you're not interested in. You're only interested in these few proteins. As we mentioned before, these transcriptional regulators, there are lots of genes, about 10% of the protein coding genes in our genome are encoding transcriptional regulators of some kind, but it's less than 0.1% of the total protein, which are these individual proteins. Yeah?
So what comes out first is going to be the one that's the heaviest protein coupled with its DNA sequence that it's bound to?
Okay, so the question is sort of a chromatography question. So in this case, we're actually, the matrix itself, the gray stuff, that is a polyionic, in fact, this case is a polysaccharide, which is just going to bind to anything that binds to DNA. So it has nothing to do with size in this case. It's just binding to that particular matrix. When we talk about size exclusion chromatography or gel filtration chromatography, which mean exactly the same thing, that's a size issue. And then the biggest one is going to come first. But in this case, it's just about the binding to whatever's in this matrix. So in this case, non-specific DNA binding proteins, and in this case, specific DNA binding proteins. But if you had a gel filtration matrix in here for the gray stuff, then it would be, you're exactly right, the big stuff coming off first and the smaller stuff coming off second. Okay, what are these transcriptional regulators doing? They're negative regulators or positive regulators. Of course, some can do both. That's why everything's in quotes here. But the whole idea of negative regulation or repressors, and this is true not just for bacteria, it's true for eukaryotic systems as well, is the binding of a repressor is going to stop transcription, and the binding of an activator is going to stimulate transcription. In the case of bacteria, it's almost always very nearby acting sequences. And so in the case of our bacterial repressors, they literally bind somewhere which blocks the RNA polymerase from binding. So in this case, the repressor is actually literally binding between the -10 and -35 sequences. Repressor comes off of the DNA, the polymerase can then bind there. Positive regulators, again, same kind of thing. Now instead you're stimulating binding by the polymerase, stimulation of binding by the polymerase when you have an activator bound, and this can be either in the presence or absence of ligand, but it's all about the binding of this activator to your DNA, stimulating the RNA polymerase being there. And the nice example of this is the lac operon, where you actually have both positive and negative regulations. So you've got repressors and activators, which are there. And originally, again, François Jacob and Monod were finding that, it's only negative regulation because they were growing everything in the absence of glucose. But here, this is sensing two different things inside the cell. The absence of lactose means that your lac repressor is binding to the DNA, blocking binding of the RNA polymerase. In the absence of glucose, you have your activator binding to the DNA. But if you have no glucose and no lactose, you're still not going to get binding by the polymerase and getting transcription. It has to be binding by an activator and the absence of binding by a repressor, which gives you the full activation of transcription. Yeah?
Can you clear up some of the terminology, just like operon, operator, new binding sites, promoter, just I'm not sure what's included in operon.
Okay, so the idea of an operon, now this is just true for bacteria. We don't talk about it for eukaryotic systems because usually it's one gene regulated by one set of gene regulatory proteins and one protein coding gene, whereas in bacteria it's usually multiple genes that are being controlled by one particular promoter. So the operon is going to be all of those genes which are controlled through one control mechanism. So in the case of the lac operon, it's the beta-galactosidase, but also the permease, and I think there's another protein, I forget what it is. It's like ZYA. One of them's the permease and allolactose, so it's an isomerase. But it's not really that important. But it's three genes together. They're being regulated by one promoter and one set of interactions with it. And so that's your operon, is all being regulated through one particular sequence. And so that is shown here. And the operator here, originally defined by our friends Jacob and Monod, because all they cared about was negative regulation, because there's no such thing as positive regulation. That process ended up being, the operator was just for negative regulation. People think about operators now much more in terms of both the positive regulation and the negative regulation here, because that's what's controlling the transcription of the whole operon. Does that make more sense?
So can we think of the operators where the repressor binds?
So that's okay. That's the question. Can we think about the operators where the repressor binds? That's what Jacob and Monod thought an operator was. I would like to think, and I'll try not to use this on an exam, but in terms of thinking about the process, it's an operon. It's regulated by particular sequences. So the operon is definitely a DNA sequence, like an enhancer would be or a silencer would be. So it's a DNA sequence that something's binding to. And so in this case, I like to think of the operon as including all of the regulatory sites which are there. And I think most people would agree that's the process. The problem is, historically, this is the only place that we have anything that's happening for regulation, so it must be the operator. Had to do with the mutational analysis which they did at the time. And if I were teaching molecular genetics, we'd talk all about it, but I'm not doing that next term. I'm doing mutant viruses from hell instead. It's much more fun. Although they're going to get as many funny accents. In bacteria, it's almost always a direct protein-protein interaction which is involved here. Either the repressor blocking binding of the polymerase or the activator helping the RNA polymerase to bind. Again, that's true in bacteria, probably because they have very small genomes relative to most eukaryotic genomes, and the genes are packed really closely together. On the other hand, eukaryotes, we've got this extra DNA, all this extra floppy stuff. So we can get away with having things acting at a distance. And so how is that acting at a distance? It's through DNA looping. And the important thing here is actually it's nice to have the model. This particular model is pretty rigid. We can't make too many bends here in our 10 nucleotides. But as soon as this would be, as long as it gets up to the ceiling, that's going to be pretty floppy. And so that's the whole idea here, is that these different binding sites on your, start again here, different binding sites on our DNA, extra red dot there, with the red dot and the white dot can interact with each other through this looping process. But it can only work if it's far enough away that you can actually bend the DNA. And we looked at some cases where we actually have specific DNA bending proteins that will help this, that will bend it really quite tightly. But if it gets too far away, and this is literally more than about 10,000 nucleotides away, there are a few examples that stuff works further away than that, but hundreds of thousands of nucleotides, you're very rarely going to have these interactions happening, but tens of thousands you definitely can. And so it's about 500 is optimal as far as is concerned. And so we talked a little bit about insulators and separating the loops of DNA relative to each other. There was just a publication that I didn't have a chance to include in the lecture where they take cells, particularly nuclei, slice them into really thin sections and sequence all the DNA in each of those thin sections, and that tells you what DNAs are next to each other. So if you chop up your nucleus into really small pieces and then sequence that DNA, it tells you what DNAs are next to each other. So it actually tells you how these things are interacting with each other. So where each of the enhancers are relative to the promoters. It's a really fascinating technique, and again, has to do with the fact that we can do ridiculous amounts of sequencing here. So this looping, that's what's happening. The dashed bits here are, again, up to tens of thousands of nucleotides. You have all of these different DNA binding proteins, regulatory sequences, which can again be enhancers or they can be silencers. Enhancers are going to bind to gene activator proteins. Silencers are going to be binding to gene repressor proteins. They're almost always going to interact with co-regulators. These are going to be co-activators, co-repressors. All comes together in this ginormous complex, and depending on how many activators, how many repressors, how much of a particular signaling molecule you'll have, sometimes the gene's going to get turned on and sometimes that gene is not going to get turned on. And we're still trying to piece apart how all of these things are regulated relative to each other. We do know that some of these activator proteins interact with parts of the general transcription factors, very much in the case of TF2D. TF2D, transcription factor for RNA polymerase IID, which contains the TATA box, but a whole bunch of TATA box associated factors. Many of those TATA box associated factors interact with transcriptional regulators. And so that's how many of them were actually found in the first place, through these kinds of interactions here. You can also have activators that will interact with the mediator. You can think of the mediators being basically kind of one of these general transcription factors. We didn't talk about it at that point, but it really falls in that kind of category. That could be interacting separately, A, D, B, E, F, H, etc., in terms of getting transcriptional initiation. In some cases, all of these guys seem to come together as what's called a holoenzyme construct, or all the pieces coming together. This is analogous to the case in bacteria. We've got the sigma factor together with all of the rest of your polymerase, and you're just bringing that in. So instead of the start with D, then have B, then have E, F, and H, and then finally get the polymerase, you can have the whole polymerase brought in at once together with B, D, E, F, and H. So it's just there are different processes and how this is working. So this is what we know about how you can start transcriptional initiation, how these activators work. Probably most of the time, your first binding of an activator, all it's doing is trying to get binding of another activator and a coactivator, et cetera, et cetera. So most cases, it's going to be this regulator-regulator interaction, which finally will get the polymerase to the promoter. That polymerase at the promoter, some promoters will start and go just by itself. In other promoters, you have to have yet another activator to get transcription to start. In some promoters, you have a polymerase which has gone a little way and stopped or paused, and then you need a transcriptional regulator. And again, these are usually complexes, multiple proteins that are involved here in order to get this RNA polymerase fully elongating and transcribing down the gene that you want to be transcribed. All of these transcriptional regulators, of course, have to deal with chromatin in eukaryotic systems. So we have, by necessity, lots of chromatin remodeling, changes that happen to chromatin in terms of getting binding of the activators and also binding of that whole pre-initiation complex. And one of the things that I like about this is it gives a little bit of an idea how big the polymerase is relative to the DNA. It's actually even considerably larger than that if you think about the whole mediator complex, et cetera. So anytime you've got chromatin-bound DNA, you're never going to have efficient binding by the RNA polymerase to that DNA. So somehow you've got to make the initiation site available. That can happen in lots of different ways, pulling the chromosomes apart with a specific chromatin remodeling complex. Again, this is an ATP-dependent process. You can have histones that get moved in and out, complete dissociation of nucleosomes, histone chaperones, or you can even have histone-modifying enzymes. And in the case of transcriptional activation, this is almost always going to be a histone acetyltransferase, so putting acetyl groups onto the side chains of your histones. What does that do? Change the charge and then allows them to come off of the DNA. Many of these histone acetyltransferases are coactivators, so they don't bind specifically to DNA, but they'll bind to your transcriptional regulators, which are bound to DNA. And so those transcriptional regulators will bring in those histone-modifying enzymes. Yeah, sure. So histone acetyltransferase. So it's putting acetyl groups onto the tails of the histones. H3 is the one that we know most about, but there are definitely modifications to all of those different histone side chains. And as we talked about before, just a second, Ian, some of those modifications are then actually bound to specifically by some of those TAFs that are associated with TF2D. Yeah?
And the acetyl groups are recognized by the bromodomains?
Acetyl groups, these acetylated histones are recognized by bromodomains. The methylated histones are chromodomains, exactly. So same thing is true if you want to shut down transcription. What's the best way to shut down transcription? Not allow the RNA polymerase to get to the promoter. What's a really good way of doing that? Shutting down the chromatin. So chromatin remodeling complexes can go both ways, as it were. They can be shutting down chromatin, causing compaction. They can also be giving it an expanded process. And that really has to do with where the nucleosomes are relative to the Tata box and your initiation of transcription. Taking off acetyl groups works just as well as putting on methyl groups. So any of your bromodomain containing proteins are going to not be associated with these histones because you've taken off those acetyl groups. Or you can have methylation and chromodomain containing proteins, which will be these nice green ones down here at the bottom, which will also shut down transcription. So questions on sort of these general aspects before we talk about a couple of specifics. Okay, again, yeah, didn't have clickers. Oh, sorry, yeah.
The recruitment of the histone deacetylase also shuts down transcription, correct?
Yes, so the histone deacetylase is going to be shutting down transcription. So this would be, and usually the histone deacetylases are not binding to DNA. So what would you call this particular protein? Would be a co-repressor, exactly. So a lot of these histone deacetylases are co-repressors. Same thing is true of these histone methyltransferases. Yeah?
If deacetyling a histone prevents transcription, what's the point of methylating it? Does that double down? Is it added?
So yeah, why bother if you can just do one? Well, the answer is probably there are two ways to skin the cat. It's an evolutionary process. But it does seem that methylation often leads to a more permanent compaction of that particular piece of DNA. And the classic example, where I wasn't planning on talking about it today, is the X chromosome. So in the X chromosome, you have massive methylation of histones, histone binding proteins. Some of those histone binding proteins are also DNA methylases, and so that really shuts stuff down. So probably, but again, it's biology. You have multiple ways to do the same thing. 4 billion years will do that for you. So yeah, let's talk about some of the specific eukaryotic regulation systems. And it turns out that this particular regulation system, one of the reasons I like it, is because it's regulated just like lac. You know what? Just like lac. It's repressors and activators. It only works when you've got activators and no repressors. And so that's what's happening here in the case of even skipped, and particularly this stripe 2 regulatory segment. And so all of these gene regulatory segments, and actually some of them are downstream. This is a little bit out of date, this particular figure here. But these are all of the regulatory segments. And each of these regulatory segments controls expression of one, turns out actually some are multiple, stripes of expression. So what does a stripe of expression mean? That means at some point in the embryo, you're going to have expression of even skipped. But all the rest of the embryo, you're not going to have expression of even skipped. And so that lack of expression is just as important as the activation of expression here. And so that's why we simplify this in terms of looking at just what's going on in this one particular place in the embryo, in stripe 2, which is regulated by this specific piece of DNA. What happens to that specific piece of DNA? Again, just like lac. It's all about binding of activators and the absence of repressors. So here, giant is going to block all of the activation here to the left of this band. To the right of it is going to be blocked by cripple, and you still have these two activators, hunchback and bicoid, so you get transcription right here. You may ask, and Ian probably is going to, why don't you get expression right here, right at the very beginning of the embryo? Because you have the two activators and you don't have the repressors there. Great question, I don't know. Maybe that wasn't your question. Whole term.
Is the only way that hunchback and bicoid, or rather giant and cripple,
Yeah, so the question here, just for recording's sake, is it just straight binding competition? That if you have the repressor there, then the activator can't bind? That does seem to be the case here. So it's a rather simple situation as compared to all the chromatin and so on and so forth that we were talking about before. So it's a relatively simple system. And actually, I now remember that the reason you don't have expression here has to do with heterochromatin, which is formed at that end of the genome. Oh, sorry, end of the embryo. Okay, so this is just again, an example of the combinatorial control. Again, absence of repressors, presence of activators, extremely similar to what's going on in lac. Now, except we have 4 of these instead of just the 2. Glucocorticoid receptor, I think this is also a nice example, one of the best understood of all of the gene regulatory proteins that have been studied in eukaryotic systems, mostly because it's really easy to turn it on or off in the presence of this hormone. The presence of the hormone, lots of different, actually, glucocorticoid hormones. Classic example is cortisol. Other ones work as well. When you have binding of this hormone, you have a change in the structure of the receptor. That then can bind to DNA. Once it's bound to DNA, it can interact with a whole bunch of other proteins, some of which are activators, some of which are repressors, and it really is much more about these protein-protein interactions or a scaffolding process, putting together this whole combination of proteins in order to get transcriptional regulation, be it up or down. Just it's really the protein-protein interactions. It's not so much the protein-DNA interactions. You have to get that protein bound to the DNA in order to get all the other ones there, but it's that interaction with all of the other proteins which is really determining whether you're having transcriptional activation or transcriptional repression, again, in response to this hormone. And this, again, getting back to your question about if you've got one way of doing it, why should we bother with any other way to do things? Again, it's probably just an evolutionary process, but it's nice with these hormone-binding proteins. Hormones, small molecules, get produced usually in some part of the CNS, can get spread through your whole body really quite rapidly, and then have different effects in different places because of all of the other proteins which are present in that individual place. It's sort of the one little match that you can light, and then will set off the rest of the fire due to all the fuel that you happen to have there. Yeah?
So under normal circumstances, you're going to transcribe at a lower level because of the co-activators and co-repressors on the left-hand side of the left graph.
So
when there's a demand for it, a greater demand for it, then you're going to release a hormone that says, quick, start transcribing more.
Yeah, so this example here, sorry to paraphrase your question a little bit. This is just specifically looking at the liver enzymes. And in these liver enzymes, you do have a small amount of expression which takes place here, but it's not true for all of the other ones. This is just these particular ones here. And then you stimulate that expression of those individual genes in the presence of glucocorticoid hormone. It's a fight or flight, whatever happens with that particular production of that hormone. But this can be very different. It could be some that are not being expressed at all under these situations, or some that are being expressed at very high levels that then get shut down in the presence of the glucocorticoid receptor. So these are just different examples here. Okay, we mentioned DNA methylation already a little bit in terms of really shutting down expression from some parts of your genome, but a little bit of DNA methylation seems to be fine. And so it's this regulation of having methylation, not having methylation. We talked about DNA methylation in terms of imprinting, how you can inherit a methylation pattern from one or other of the parents, male or female, and that can lead to differential expression in an epigenetic inheritance kind of process. The vast majority of methylation that happens in our genomes is 5-methylcytosine, right here. 5-methylcytosine, of course, can be a problem as soon as it's deaminated because it then looks like thymidine. Thymidine then will, uracil, excuse me. Uracil will then get chopped out of your DNA, and you end up with a mismatch, actually a mutation, which happens at this particular position. Almost all of this methylation happens where you have a CG right next to each other. That gets maintained by the maintenance methyltransferase. And all that maintenance methyltransferase does is said, hey, we've got a hemimethylated piece of DNA. As soon as that gets replicated, we'll add one. If you have a part of your DNA which is not methylated, it's not methylated usually because you have binding of some other protein to that particular CG sequence, particularly if you've got promoters or anything like that. That's where you see high concentrations of CGs. If you have methylation, that's usually going to be a difference in terms of what's binding to that particular position. So we already talked about how we get our RNA. Now the question is, how do we end up with protein synthesis? So it's basically, how do we go from our transcriptional initiation, all these next steps through the whole process? We talked a little bit about premature termination of your transcription. That's your attenuation or abortion of your transcript. That happens with riboswitches, where you have a change in the structure of your RNA giving you a terminator. Capping can be regulated. We haven't talked about that in too much detail, but there's plenty of that. Alternative splicing, lots of alternative splicing is happening. You can have splicing activators, splicing repressors, sex-lethal and transformer are the examples we talked about there. RNA editing, again, very bizarre mechanism. Why the heck would you edit your RNA when you could just mutate it and have a different gene? Seems to have worked, so don't mess with it. This is true for, again, some developmental genes, brain development, etc., very specific changes that happen. Usually it's by a specific enzyme that binds to a specific structure that forms in your RNA, but also a viral defense mechanism deals with lots of modifications to RNA, editing of the RNA in a hope, to totally over-anthropomorphize here, of such mutation of your virus sequence that it can't be functional anymore. Nuclear exports, this has also been taken advantage of, particularly by RNA viruses, to make sure that the whole RNA of the virus gets exported, so it avoids the whole process of exon junction complexes and so on and so forth. Once the messenger RNA gets out, you can put it in various different places. This is exactly how you get the bicoid and hunchback gradients in the Drosophila embryos. All those messenger RNAs get put at one end of the egg. As soon as it gets fertilized, those messenger RNAs get translated, and you end up with a gradient of proteins starting at one end. We looked a little bit about translation. Starts, particularly in bacteria, has to do with access to the Scheindel-Garnel sequence, which is where the small subunit of the ribosome is going to bind to, and then allow translation to take place. My favorite of those is the RNA thermometer. Again, forms a secondary structure at one temperature. As soon as you heat it up, that structure is completely gone. Sort of the simplest mechanism of regulation that I could think of. And then we talked a little bit about RNA degradation. I'm going to talk more about that now. So this is review of George's lecture. Just pulled out a couple of things in there. The first one are these internal ribosome entry sites. This is, again, a structure in the RNA which forms a structure in your messenger RNA that allows you to get translation to take place in the absence of a cap structure. So internal ribosome entry sites. Yeah, people thought it was just the viruses, but it turns out there are quite a few cellular genes that do this just as well. And the big advantage here is you don't necessarily need all of these translational initiation factors. Turns out there are different irises those different irises require different translational initiation factors. But the main thing here, just as is true with regular translation, where it's cap dependent, is getting the small subunit of the ribosome to the RNA and to the translational start site. So here, iris allows you to get the small subunit of the ribosome together in an iris-independent process. It's the cap and the cap binding that allows you the small subunit of the RNA to get on there. Regulation of translation in eukaryotic systems with iron regulation, something that I think George forgot to mention. Why do we care so much about iron? Any ideas why we care so much about iron? You heard about the Fenton reaction? No? Chemistry is a different class. Why am I thinking about chemistry and molecular biology? So one of the chemistry professors always gives me a hard time when he sees me carrying this around. He's like, "You're teaching chemistry, aren't you?" But yeah, we're not going to get into all the details about the Fenton reaction, but basically, anytime you've got iron or too much iron inside the cell, you start making reactive oxygen species. And reactive oxygen species are a huge problem for DNA, for proteins, for membranes, etc. So regulating the amount of iron that you have inside the cell is really, really important. And that's probably why this mechanism has evolved. So what ferritin does is ferritin will bind to bind to DNA. Back up. Ferritin binds to iron. Been talking for too long here. So ferritin binds to iron, and what it does is it sequesters the iron. Actually kind of makes a cage around the iron so it can't react and can't produce oxygen species. So if you have too much iron, you want to make this cage that will take care of all the iron. On the other hand, a lot of our enzymes need iron in order to be able to function. So you need a very limited range of iron. If there's too little iron around, you need to get a bunch of iron inside the cell. Too much iron around, you need to protect yourself from all of that iron. So transferrin is getting the iron in. Ferritin is, if there's too much iron, getting rid of it. And so the way that works is at a messenger RNA level. It's the same enzyme that regulates both iron sequestration, hiding it, and bringing iron in, and just has to do with where this secondary structure is in your messenger RNA. If it's at the 5' end binding to aconitase, you don't get translation, no ferritin is made. If it's at the 3' end, it stabilizes the messenger RNA, and you get this messenger RNA made and translated, made into a transporter. Presence of iron, too much of it, you want to make a sequestration. So now you allow translation. Here, you don't want to bring any more iron in. We're already trying to make ferritin to get rid of it. So now this comes off of your messenger RNA. That messenger RNA gets degraded through the regular 5' to 3', 3' to 5' exonucleases. And in the case of the transferrin receptor, I think there's also some specific endonuclease activity that takes place here as well. So mostly what George talked about with the small RNAs, RNA interference in particular, but the whole idea here is you have a double-stranded RNA, relatively long double-stranded RNA, that then gets cut into a smaller piece of RNA, and that smaller piece of RNA will interact with what you're trying to regulate. Now, there are really two different flavors of these. Actually, there are multiple ones, but the main ones we'll talk about are the microRNAs and the small interfering RNAs. Big difference between these two is the microRNAs are what I like to call endogenous RNAs. These are transcribed from genes in the genome. So your genome is encoding these small RNAs. And actually, they should probably update this. Actually, probably thousands of genes for these things. So these microRNAs are transcribed normally by RNA polymerase II. They've got caps on them. They got tails. But the big deal is that they're self-complementary to each other. So they will form these double-stranded RNAs that then get chopped up into short RNAs that can lead to degradation or repression of translation. And that just depends on how much base pairing you happen to have here. Small amounts of base pairing, less translation, lots of base pairing, RNA degradation that happens. So these are endogenous. These are genes, regular parts of your cellular genome. On the other hand, most of the small interfering RNAs are exogenous RNAs. So they've come from outside. Now that's a little bit of a exaggeration because, of course, transposons are already in your genome and centromeres are already in your genome. But certainly transposons probably came originally from outside the genome, and they're not specific single copy genes that you have for these things. And centromeres also, you've got many copies of the same sequence, which is there. So what happens is you end up with double-stranded RNA, long double-stranded RNA, just from individual RNA molecules. So it's not just one that's base pairing back in itself. It's two RNAs that can base pair with each other. Why most viruses make these RNA viruses, we'll talk all about next term, when you have to listen to me again if you're taking virology. These then get chopped into smaller pieces and again go into these protein complexes, which can either lead to degradation of the RNA, which makes a lot of sense, as if it's a viral infection. You want to chop up that RNA in such a way that it can't actually infect the cell anymore. Transposons and centromeres, on the other hand, also have lots of double-stranded RNA. This is background transcription. Almost the whole genome is being transcribed all the time at low levels, but being transcribed all the time. Anytime you've got copies of identical sequences, those are going to form double-stranded RNA, and usually those lead towards RNA-induced transcriptional silencing. So it's a double-stranded RNA that forms that blocks the RNA polymerase, which makes a lot of sense. If it's a transposon, you don't want transposons jumping all over your genome all the time. So you want to shut down the transcription of them, or if they do get transcribed, chop up that messenger RNA such that the transposon doesn't make the transposase and is jumping around in the rest of the genome. Yeah?
And that's what RISC does?
So the RISC pathway chops things up. The RITS pathway just silences. Okay, more questions on those small RNAs? The other small RNAs we talked about come from CRISPRs, the clustered regularly interspaced palindromic repeats. No, I'm not going to ask you what it stands for on the exam. But these are regions in the genome, and if I'd given this lecture, I would have shown you a picture of the guy who actually initially discovered these, a fellow by the name of Yoshi Ishino. But he just found these really strange repeated sequences, all these orange ones, present in the E. coli genome. It's only much later that they found the pieces of virus-like sequence between these repeat sequences. And then, even later, literally now, about 10 years ago, showed that this was important for blocking virus infection. So George went through the three processes here that are involved in protecting the cell from a virus infection. The first one is when you have a virus infection that takes place, how do you get that into your repeated sequence, this whole set of repeats? How do you get this sequence in between some of those repeats? That's Cas1 and Cas2, which bind to these sequences if they have a protospacer adjacent motif. And this is the way that you can tell that an incoming DNA is not your own DNA, because if the Cas9 system was chopping up all your CRISPR loci, you'd be chewing up your own genome. That would be completely counterproductive. So when a new DNA comes in, presumably from an infecting virus, it's this protospacer adjacent motif that says, hey, chop a piece of this genome out, put it into our CRISPR locus so we can be resistant to this later on. How does that resistance take place? These, again, are these repeated sequences with virus sequences and the spacers in between them. There's also a leader sequence in all of these CRISPR-Cas loci. This leader sequence is a really nice promoter. So what it does is it will now transcribe this whole set of genes with repeat sequences, spacers, repeat sequences, spacers. Repeat sequences, since they're palindromic, love to form secondary structures, and so that's what this secondary structure is here. This then gets chopped up by some of the other Cas proteins that we haven't talked about. RNase 3, in the case of CRISPR-Cas9, you have this short RNA, which has mostly spacer and a little bit of your repeat sequence, together with the tracer RNA. That then interacts with Cas9 in order to allow cleavage of any new DNA, which is matching your CRISPR spacer, which is shown up here. How does that process take place? Again, your CRISPR spacer here. It undergoes strand invasion, really similar to what you see with double-stranded break repair, actually. So Rec-A mediated, getting that single strand into your double-stranded DNA. And then cleavage takes place, and not at all surprisingly, some of that cleavage is in a very similar way to what you see in holiday junction resolvases. So this RUV-C is a holiday junction resolvase. And so this is a part of your protein, the Cas9 protein, that's very similar to RUV-C and has a similar activity. We'll cut the DNA. It's different with the resolvase here, as it doesn't ligate it back together. It just leaves it once it's cut. Yeah?
RUV-C is similar to what?
RUV-C is similar to a holiday junction resolvase. So holiday junctions are where you have that crossed over DNAs, two strands coming into each other. Those then get cut by RUV-C. Okay? And then in the case of the resolvase, it would religate them. This just does the cutting process. So you end up with a double-stranded break. Those double-stranded breaks get repaired somehow. Originally, it was by non-homologous end joining. So you had some cleaving into the ends, and those then get put back together. The more advanced techniques now with CRISPR-Cas9 use homologous recombination in order to do that particular genome editing. So I've got a couple more things with proteins and DNA. Do you want me to try and go through these, or shall we call it quits and go from there? Keep going. Okay. So I'll talk fast. Column chromatography, we kind of talked about this already when we talked about the affinity chromatography. It has to do with the matrix slowing down things coming off of your column. SDS page, important thing here, you're separating in an electric field. Most proteins have incredibly diverse kinds of charges on the outside. To change that, you need to bind them to some kind of ionic detergent. Here, SDS. They will now be separated based on size. Western blots, there's some confusion about southerns and northerns and westerns. Westerns is all about proteins. So now you have a protein gel. This could either be an SDS page gel or a 2D electrophoresis gel. This then gets transferred to a membrane, and once transferred to that membrane, you can detect the proteins with antibodies. It's the blot down here at the bottom. Gene to protein, we'll finish here. We talked a lot about genes. We've discovered these. You can actually take this sequence, compare it to what you found in the database, and say, oh, okay, I think I've got an idea what this does. You can use what we talked about on Wednesday to take this DNA, PCR, or cloning by just taking the piece with your restriction endonucleases, make a whole bunch of your protein, figure out what that does. Or if you have a new protein, you can figure out what that sequence is, usually with mass spectroscopy. And then because we've got all these genome sequences, you can find where it is in your genome. You can make the DNA with PCR and go through this whole process again and again. Nucleotide electrophoresis, it's all about the detection. So we spent all this time talking about radioactivity. Ethidium bromide. Blots, I just realized that this is not in our current version of our textbook. Sorry about that. old school stuff, I guess. Everyone does sequencing instead, but it's all about finding the individual piece that you were looking for. So back here, for instance, you want to know if this is the piece that has your favorite gene in it, and then you could use that one to clone, or this piece down here has your favorite gene in it. Through that hybridization process with that short probe that we talked about making, that's the way you can identify it on a blot. And DNA sequencing is totally cool. Good luck at the exam on Wednesday.